Hello everyone. InformationBoxTicket Lifestyles brings you today biotechnology topic on plant tissue culture. But first, don't forget to like this video, subscribe to my channel and press the bell icon button so you don't miss any of my videos. The table of content includes plant tissue culture medium, role of plant growth regulators, types of cultures, plant tissue culture procedure, plant regeneration pathways, and lastly, applications of plant tissue culture. Let's start with a brief introduction of plant tissue culture. Plant tissue culture is an in vitro technique in biotechnology that is generally used for aseptic isolation and culture of single cells, protoplasts, tissues, organs, and their component on artificial media under aseptic conditions so that these parts can grow and develop into complete plants. The theoretical framework of plant tissue culture is based on the unique property of cellular totipotency and regeneration. Cellular totipotency is the capacity of the plant cell to regenerate into entire plant. The experimental evidence of totipotency was first time obtained by Gottlieb Haberland in 1902. Haberland grew pulsite cells from leaves of various plants, Lamium, Propurium, Pulmonaria, Eutrica, etc., in Kopp's salt solution in hope of regenerating whole plant from it. The cells increased in size, survived for several months but failed to divide. Later, the cultures were lost due to microbial contamination. On the basis of his pioneering experimentation, Haberland is credited as the founder of plant tissue culture. After that, during the period 1902 to 1930, attempts were made to culture isolated plant tissues and organs such as the root, shoot, apex, and embryos. During this period, different types of nutrient media were evaluated for the culture and differentiation of organs and tissues. Lots of experiments were carried out to formulate suitable nutrient media and standardized culture conditions, including various plant hormone combinations on in vitro morphogenesis. Next, plant tissue culture medium. Plant tissue culture medium contains all the nutrients required for the normal growth and development of plants. It is mainly composed of macronutrients, micronutrients, vitamins, other organic components, plant growth regulators, carbon source, and some gelling agents in case of solid medium. Uracetin and Cox media, MS medium, is most extensively used for the vegetative propagation of many plant species in vitro. The pH of the medium is also important that affects both the growth of plants and activity of plant growth regulators. It is adjusted between 5.4 to 5.8. Keep showing your support to this channel by subscribing to it. Role of plant growth regulators in tissue culture. Plant growth regulators play an essential role in developing the development pathway of plant cells and tissues in culture medium. The type and the concentration of hormones used depend mainly on the species of plant and the tissue or organ cultured. Auxins and cytokinin are most widely used plant growth regulators in plant tissue culture, and their amount determines the type of culture established or regenerated. The high concentration of auxin generally favors root formation, whereas the high concentration of cytokinin promotes shoot regeneration. A combination of both auxin and cytokinin leads to the development of mass or undifferentiated cells known as callus. Let's go into the types of cultures. Tissue cultures or micropropagation may be: number 1, organ culture; number 2, explant culture; number 3, callus culture; number 4, cell suspension culture; and lastly, number 5, protoplast culture. Let's learn about organ culture. Culturing isolated organs in an artificial media under controlled conditions are known as organ culture. The following are the various types of organ culture and its specific purpose. Seed culture. It increases the efficiency of germination of seeds that are difficult to germinate in vivo. It involves the precoccination, germination by application of plant growth regulators, and production of clean seedlings for explant or meristem culture. Next is embryo culture. It helps to overcome embryo abortion due to incompatibility barriers, overcome seed dependency and self-sterility of seeds, and embryo rescue in distant hybridization where endosperm development is poor. It also helps in shortening of breeding cycle. Ovary or ovule culture. Ovary is a common explant for the production of haploid plants. Ovary culture helps in overcoming abortion of embryos of wild hybrids at very early stages of development due to incompatibility. Anther and microspore culture. Anther and microspore culture is used for the production of haploid plants. Production of homozygous diploid lines through chromosome doubling, thus reducing the time required to produce inbreed lines, and it also helps in uncovering mutations or recessive phenotypes. Number 2. Explant culture. Culturing of any excised tissue or plant parts such as leaf tissue, stem parts, cotyledon, hypocotyledon, root parts, etc. is called explant culture. The primary purpose of explant culturing is to induce callus cultures or to regenerate whole plantlets directly from it without the formation of callus. Short apical meristem culture is an example, and its important uses are production of virus-free germplasm or plantlets, mass production of desirable genotypes, cryopreservation, cold storage or in vitro conservation of germplasm. Number 3. Callus culture. Callus represents an unorganized or undifferentiated mass of cells. When an explant is cultured in a medium supplemented with sufficient amount of plant hormones, it starts producing mass of cells from the surface of the explant. Callus cultures can be maintained for a very long time by intermittent subculturing to a fresh medium. Callus cultures can be used for regeneration of plantlets, preparation of single cells or suspension cultures or for protoplast preparation, genetic transformation studies, and lastly, the generation of useful somoclonal variants. Number 4. Cell suspension cultures. Single cell cultures and suspension cultures can be established from callus cultures by transferring a piece of callus tissue into liquid medium and subjecting it to continuous shaking. The rate growth of the suspension cultured cell is generally higher than that of solid culture. By subculturing for several generations, a fine cell suspension culture containing small cell aggregates and single cells is established. The cells in suspension are also used for a large-scale culture with jar fermenters and tanks. The suspension cultures can be grown either as batch cultures or as continuous cultures for producing phytochemicals in airlift bioreactors. The cell suspension cultures can be used for inducing somatic embryogenesis and the preparation of artificial seeds, induction of somatic mutation, and selection of mutants by screening the cells just like microbial cultures. The cells can also be used for genetic transformation experiments to produce transgenic plants. And lastly, number 5, protoplast cultures. Protoplasts are plant cells without cell walls. The cells may be taken from the leaf tissue or from any other part of the plant or from the suspension cultures. These cells are incubated in an enzyme mixture containing of cellulase, hemicellulase, and pectinase for a specific period of time. The enzyme mixture acts on the cell wall and is completely digested, so that the underlying cell membrane is exposed. The plant protoplast can be used for various biochemical and metabolic studies. It can be used for somatic cell fusion to create somatic hybrids, Formation of cybirds, in which there is no fusion of nucleus but fusion in between the cytoplasm. Protoplasts can also be used for genetic transformation studies with biolistic methods, electroporation techniques, by PEG-mediated DNA transfer, or by direct injection of DNA into the nucleus of the protoplast using microsyringes. If you wanted to see more lectures related to biotechnology, don't forget to subscribe. Plant tissue culture procedure. The following are the major steps involved in plant tissue culture. Number 1. Selection of explant. Number 2. Surface sterilization of explant. Number 3. Initiation of culture. Number 4. Shoot formation. Number 5. Root formation. And lastly, number 6. Hardening. In the first step, selection of explant, leaves, stems, or any other suitable plant parts may be cut from healthy plants. The explant should be cut to suitable sizes. Old tissue plants should be avoided. Wash the dust off the tissue in a beaker of distilled water, which should be surface sterilized. In the next step, surface sterilization of explant. This part of the procedure should be carried out in a laminar flow hood cabinet. The leaf or any other tissue should be immersed in 70% ethanol for 30 seconds and then transferred to a sterilized petri dish. Sterilized tissues and forceps are then used to cut the tissues into small suitable sizes of explants. The explants are transferred into a 10% hypochlorite bleach solution or mercuric chloride for 5 minutes, gently aggregating once or twice during this time. They are then washed thoroughly by immersing in sterile distilled water for 2 to 3 minutes. Step 3. Initiation of culture. The surface sterilized explant is implanted into a suitable nutrient medium with a correct type of growth hormone in appropriate concentrations. Inoculated cultures are transferred to growth chambers or tissue culture rooms having appropriate physical conditions such as the right temperature, 26 to 28 degrees Celsius, relative humidity 50 to 60%, and fluorescent light, 16-hour photo period. Depending on the nutrient conditions and hormone combination, the explant either starts developing callus or directly produces shoot buds or roots to form a large number of plantlets. For the shoot formation, the healthy callus are transferred to shoot induction medium having cytokinin for shoot initiation, whereas for root formation, after development of shoots from callus, they are excised and transferred to a rooting medium having auxins. Shoots after development of roots are ready for being transferred for the greenhouse for hardening. Step 4. Transplantation and hardening. The plantlets that develop roots are transferred to a special pots with sterilized sand for hardening under greenhouse conditions. Hardening is a process by which the in vitro generated plants are accumulated to a greenhouse conditions. Then they can be transferred to the field. Don't forget to press the bell icon button so you don't miss any of my videos. Plant regeneration pathways. There are two methods of whole plant regeneration. Number 1. Organogenesis. Plant regeneration through the formation of shoots and roots is known as plant regeneration through organogenesis. Number 2. Somatic embryogenesis. The cells under a particular hormonal combination changes into zygotes, somatic zygotes, and follow an embryonic path of development to form somatic embryos. These somatic embryos are similar to normal embryos and can develop into a complete plant. Applications of plant tissue culture. This part of the lecture will be continued in the part 2 version of plant tissue culture. So thank you so much for watching till the end. Don't forget to like this video, subscribe to my channel and press the bell icon button so you don't miss any of my upcoming videos. Thank you so much.